Hello students, our chapter is molecular basis of inheritance. In that we study about the DNA packaging. In this DNA packaging, in previous lecture we deal with the packaging in prokaryotes. You know it very well about the prokaryotes. What is meant by prokaryotes? All those things we are deal with the in previous lectures. Now today we deal with the packaging of eukaryotes. Packaging of what? Packaging of DNA in eukaryotes. And first question is, what is eukaryotes? Right? What is mean by eukaryotes? Eu means true. Here eu means true. Karyon means nucleus. The true nucleus is present inside the cell. That cell is called as eukaryotic structure. That structure is also called a eukaryotic structure. Understand? What is eukaryotic structure? That eukaryotic structure having his own nucleus. This is the nucleus. Then that nucleus is well organized. What is that? That nucleus is well organized nucleus. Well organized nucleus means that nucleus having nuclear membrane. Then that nucleus having nucleolus. Here is nucleolus. All these presence of all these nucleus, nuclear membrane and nucleolus, because of that this nucleus is called as well organized nucleus. Then inside the nucleus we found a huge network of genetic material, or which is called as a chromatin network. What we call that is a presence of chromatin network. Where is chromatin network present? Chromatin network is present inside the nucleus. Also we can call it as a chromosomes. We call it as a chromosome. The chromosomes, they are in the form of chromosomes. They are condensed. Chromosome is condensed when they are participate inside the in all cell division activity, that is meiosis, mitosis. You can study, you will also study in the previous lecture, that is cell division chapter. In that cell division chapter you discuss about the meiosis and mitosis, and inside that meiosis and mitosis we found a condensation of chromosome for the purpose of cell division. Then the DNA is associated with histone and non-histone. Here this chromosome, then chromosome is also this chromatin network is also, we can also call it as a DNA. What is DNA? DNA is also genetic material. With chromatin network or chromosome, we also call it as a genetic material. This DNA also we can call the genetic material. Then what is genetic material? Genetic material is that material which transfers genetic information from generation to next generation. Then DNA associated with histone and non-histone proteins. DNA is associated with to whom? To histone and non-histone proteins. Right? Here, then, this concept is reported by R. Conver in 1974. Right? Then this histone is bind tightly to whom? Bind tightly to the protein, bind tightly to the protein, right? Then this non-histone is a release of DNA. They are opposite to each other. They are doing work opposite to each other. When this histone is binding tightly, that the non-histone is release that DNA from the binding. When at the time of DNA replication, then this DNA replication is done by this non-histone protein. Then DNA histone, non-histone, and all these three accumulate together to form a chromatin material. It is, and how that chromatin material? It is complex nucleic acid. It is complex nucleic acid. And what is nucleic acid? That is DNA and RNA. DNA means deoxyribonucleic acid, RNA means ribonucleic acid. And also the supporting is proteins for formation of nucleic acid. Also called DNA is a blueprint of our body. Then this concept is very simple. What? A blueprint. When we saw any plant, a seed of a particular plant, then after the germination of that particular seed, we found a similar plant. We observed a similar plant is grow there. That is that concept, and that character is transferred from his DNA. That's why this DNA is also, we call it as a genetic material. Then our next point is, that is a nucleosome. Here nucleosome. And this nucleosome play a very important role for what? Formation of the DNA, or to formation of a supercoiled structure of a DNA. Right? Here, for that, we need to understand all those things present in this nucleosome. We are present in the nucleosome. What is nucleosome? What is meant by nucleosome? Nucleosome is a part of nucleus. Here we study about the nucleus, and it is a part of nucleosome is present inside the nucleus. That's why it is we call it as a nucleosome is called as a part of nucleus. Then we can take details about this nucleosome. How this nucleosome formed, who is responsible to forming this kind of structure. Start from the histone proteins. Here these are the histone proteins. These histone proteins, there are four types. The four types of histone proteins molecules, they are positively charged. Okay, they are positively charged, and they are also called as amino acid molecules. Okay, then these positively charged particles, or the positively charged histone proteins, they are binded by this, or they are wrapped by this negatively charged DNA. Here you can observe these are that stand. They are bind these positively charged structures. And how they the four types of that histone proteins are H2A, H2B, H4, H3, and they are bind to each other and to form a 8 molecule structure, and that 8 molecule structure is called as octamer of histone. Octamer of histone. Why we call this atom here? Because it contains 8 molecules inside that. Here, one protein, this H1, this one protein, that is H1 histone protein, is also play a very important role to bind this structure, to formation of this nucleosome. Then this nucleosome, how this nucleosome do function like this? One nucleosome contain 200 base pair. One nucleus contain 200 base pair, repeated unit of chromatin. Repeated unit of chromatin. In previous lecture we study about the chromatin. What is the chromatin? Chromo means color. Chromatin material, that is a transfer of color characters from one step to another. The nucleosome core is made histone protein, right? Which type of histone protein? They are H2A, H2B, H3 and H4. These are the binder. Okay. Then the H1 protein bind here. Here is the H1 protein. Then the DNA thread where it enter and leave the nucleosome. The DNA is enter inside and leave the nucleosome. The DNA present in each nucleosome. DNA is present in each nucleosome. Here this DNA is present. Where? In each nucleosome. Here this is a combined unit of nucleosome. It is repeated unit of chromatin which are thread-like. They are repeatedly formed. Here, if this is one nucleosome, then here we found another nucleosome. Here again we found another nucleosome. These are the repeated unit of this nucleosome. It is repeated unit of chromatin which are thread-like stained. Thread-like stained. Stained means colored. And this color is about what? Color is hair color, skin color and eye color. That is the stain, we can call it as a stain bodies present inside the nucleus. Right? This looks like a bead on string. Bead on string. Like this. These are the beads on the string. These are the nucleosome and this is the DNA. That's why they are looking like a beads on string. When observed under when they look like this, when we can observe it under the electron microscope. DNA helix of 200 bps wrapped around the histone octamer by 1/3/4 by 1/3/4 turns. DNA wrap this nucleosome and histone protein about how many turns? 1/3/4 turns give that to wrap this histone protein. Then 6 such a nucleosome get coiled and then form a solenoid. Looks like a coiled tenifolic wire. They are looking like a coiled tenifolic wire. The chromosome packed to form a solenoid structure that is 30 mm in diameter. We can observe in our next diagram. We have also another diagram here. In this diagram, we can discuss about how this chromosome, this chromosome size of the chromosome is 400 nanometer. This size of chromosome is 400 nanometer, and that 400 nanometer is combined in our small nucleus. That's why we need to coil that particular structure. That is in the form of supercoiled structure. Then here our chromosome is 400 nm. That chromosome, they are coiled and they are formed a protein loop. Protein folds are formed by this chromosome. Then after that they form a 30 nanometer folds, and this diameter is 30 nanometer, and we observe this single beaded structure we found here, then another structure, then 4 cell structure of histone protein are observed here. These are the 4 cell structure like this. These 4 cells, they are bind by the DNA itself, and here the group of 8 histone protein, they are combined here. That is, in previous one we called it as a nucleosome, and like this they are formed coiling, and here also we found a H1 protein attached to linker DNA and nucleosome. H1 protein attached to whom? To linker DNA. H1 protein. In previous diagram we studied about that H1 protein, and this what is meant by this linker DNA? Linker DNA is that DNA which connect that nucleosome with each other. This nucleosome is attached or linked by this DNA. That's why we call it as a linker DNA. Then after that, coiling process, completion of coiling process, that 1400 nanometer chromosome, they are forming a supercoiled structure, and they are forming a supercoiled structure inside the nucleus. Then anything remain, that is the packaging of chromatin at higher level needs additional set of protein that are called as non-histone chromosomal protein, that is called as NSC. These are additional set of protein that contribute to the packaging of chromatin at higher level, that is heterochromatin and euchromatin fibers. We can take a study details about that heterochromatin fibers. The heterochromatin in eukaryotic cell, some segments of chromosome, that during interphase, cell division, we found prophase, metaphase, and in the cell division we found interphase, and interphase we found early prophase remain in condensed state. This region constitute heterochromatin. This region contain a heterochromatin. This term was proposed by Hayes. The name of scientist, that is Hayes. These regions are localized near centromere. Where? Near the centromere of the chromosome. Here the centromere. And all these are present near the centromere. Then the centromere, telomere, and also interconnected. Heterochromatin is genetically inactive. Heterochromatin is genetically inactive, and non-histone chromosomal proteins, that is NSC. This non-histone chromosomal protein having two types, that is heterochromatin and euchromatin. This heterochromatin and euchromatin, they are different from each other. How they are different from each other, how they are working from each other, look here. Here, the during interphase and early phase, this heterochromatin is observed, or we can observe in a condensed state, where in term of in interphase and early prophase. Who proposed that? Then the scientist, his name of scientist is Hayes. He proposed that. Localized near the centromere, where this heterochromatin localized, they are localized in centromere, telomere and intercalculated. Then genetically they are most inactive. The stain are strong, that's why they appear dark in color. Then they are 2-3 times more rich in DNA than euchromatin. They are 2-3 times. Here we found a DNA is present there, and the nucleosome, they are bind by this DNA, and this is a clump of, they are present in the form of clump, that is heterochromatin. Then next is euchromatin. Euchromatin is the rearrange of chromonema, which are in non-condensed state. Here they are the non-condensed state. This is the condensed state. Here are the non-condensed state. Stay light in color. Genetically, it is very active. Here genetically most inactive. Genetically very active in transcriptional process. This is all about the non-histone protein, non-histone types of non-histone chromosomal protein. Then these all things we are discussing, all points we are discussing, and what is that? That is this is our chromosome. This is also chromosome. This is our chromosome. This chromosome, the size is 1400 nanometer. Right? Size is 1400 nanometer. And this is condensed in and to add inside the nucleus. That's why nucleus is smaller. That's why we need to form a superchromatin material, superchromatin network. And here we found nucleosome, DNA. Again they are condensed. This is the DNA. They are condensed. Again they are condensed to form a supercoiled structure, and this supercoiled structure, they are converted into 2 nanometer. They are converted into 2 nanometer. This is the main thing about the DNA packaging. Okay, thank you.